your glycolysis and Krebs cycle is your acetyl CoA. Okay, so this is a product which is being formed through the link reaction, and this link reaction is forming what? They are forming. So this is one pyruvic acid or the pyruvic molecule which is undergoing the oxidation. As we have discussed, there is formation of two pyruvate. Okay, so ultimately, if we talk about the link reaction, okay, we are having two NADH which have been formed because there are two pyruvate. And there will be also formation of 2-acetyl CoA. So by the end of link reaction, if we talk about, we are having 2 NADH and there will be formation of 2-acetyl CoA. So that is actually the product of when you're talking about link reaction with the liberation of your CO2. That is by the process of oxidative decarboxylation. Okay. So this is the product that has been formed by means of link reaction. Why we are calling it to be linked? Because it is joining the product of glycolysis to the Krebs cycle. That's why we are saying it to be linked reaction here. Okay, so this is now this acetyl CoA. Now we are coming to the main topic here, that is acetyl CoA. You can see here that acetyl CoA is being formed. Now we are coming. Is this part okay to everyone?
Yes, sir.
Yes.
On the blackboard, if you will write bigger letters, it will be visible. Right now, what you are writing is not visible on the screen. It is not clearly visible on the screen. If you will write larger letters, will increase the size of the letters, it will be visible. Okay. Stepwise again write larger letters, it will be visible to us.
Your link reaction, by the end of link reaction, what we are saying, there is formation of 2-NADH plus H plus, and there is also formation of 2-acetyl CoA. Okay, this is the product which has been formed by the end of, that is your link reaction. So during the process of link reaction, as I mentioned, the pyruvic acid or the pyruvate, it is undergoing oxidation with the release of your carbon dioxide. So it is your oxidative decarboxylation. So that is formation of acetyl CoA. The name of the process is actually oxidative decarboxylation. Okay, so that should be very clear. Okay, and it has to be mentioned to the students here. Oxidative decarboxylation. Okay, and there is formation of NADH. Okay, so this NADH, when you're talking about, they are also the source of energy. This NADH, which is the reduced coenzymes actually. Okay, so they are also the source of energy. But they will be used where? They will be used in the ETC, electron transport system, or the chain. Okay. So this is your link reaction. Now we are moving to the main part of the Krebs cycle here. Okay. So when you talk about the Krebs cycle, okay, we know that the Krebs cycle especially comes with the discovery. Sir Hans Krebs in 1937 discovered this pathway of cyclic oxidation of acetyl CoA. So in the Krebs cycle, what we are seeing, it is oxidation of your acetyl group which is taking place, and ultimately we can say that it is the oxidation of the glucose which is taking place with the pyruvic acid, where the intermediate or the linking one is your acetyl CoA. So ultimately we can say, so in the process of link reaction, what we are seeing, there is formation of one CO2 here. But when we see the equation of respiration, that is the cellular respiration, the product side we see that there is formation of 6 CO2. So this 6CO2, when we see C6H12O6 plus 6O2, it gives 6CO2. So where this 6CO2 is coming from? So one CO2 is coming through your link reaction and the other two will be coming. So this link reaction will be coming to where? This is your Krebs cycle. They are coming to the Krebs cycle. And in the Krebs cycle, we have two decarboxylation. There is a two-step which involves decarboxylation. CO2 will be given at the two steps. So two CO2 here and one CO2, 3. As it is only one we are talking, suppose it is 2 acetyl, so ultimately it will give rise to your 6 CO2. Okay, by the process of decarboxylation. So that should be also told to the students. We can link how it is 6 CO2 which is being formed here. One acetyl is giving out, okay, when there is oxidation of your pyruvate, formation of acetyl CoA, it gives 1 CO2. In the Krebs cycles when we will be discussing, there is also formation of 2 molecules of CO2 from one acetyl which is undergoing, when you're talking about acetyl CoA, which is undergoing your Krebs cycle, that is oxidation process here. So this will be your 6 CO2. Now we are seeing, especially the location. If we draw the diagrammatic representation, suppose this is the diagrammatic representation, we know this is your Krebs cycle is taking place in the matrix of the mitochondria. Okay, the Krebs cycle is taking place in the matrix of mitochondria. Okay, so this should be told to the students. But and why it is taking place in the matrix? Because the enzymes which is required for the Krebs cycle, okay, it is present in the matrix. But one of the enzymes, all the enzymes are present in the matrix, but one of the enzymes, especially the succinate dehydrogenase, they are present along the inner membrane. This is the inner membrane. This part is the inner membrane. So they are present in the inner membrane, succinate dehydrogenase, the only enzyme of the Krebs cycle which is present along the inner membrane. The rest of the enzymes, they are present in the matrix of the mitochondria. Okay, so that should be mentioned here. Now when we are beginning with the Krebs cycle here, that is the mechanism. Yes. Yes. Now we are beginning with the mechanism. So as you can see here, the mechanism, okay, we have seen through the link reaction how acetyl CoA was being formed, and this acetyl CoA which is being formed, okay, the pyruvic acid was 3 carbon molecule, but acetyl which is being formed, acetyl CoA, rather the acetyl, it is your 2 carbon molecule. The acetyl which is being formed, acetyl CoA, that is a 2 carbon. Why it is 2 carbon? Because the pyruvic acid or the pyruvate was 3 carbon, now it is here 2 carbon. So we should, I have mentioned here, it is undergoing decarboxylation. So there is release of 1 CO2, so 1 carbon is lesser. So that's why acetyl group here, okay, that is having 2 only carbon atom. And the oxaloacetic acid, okay, they
Raju sir, your speaker is muted.
Cannot hear you. Raju sir, you are not audible.
That's why it is also known as, when you're talking about tricarboxylic acid cycle. So the first stable product, or the first product of Kreb cycle is citric acid. So that's why it is known as citric acid cycle. Okay. So this has to be told to the students here. Okay. First product, and this is being catalyzed by an enzyme that is citric synthase. Okay. So this is the first reaction of your Kreb cycle. Now this citric acid, okay, will be undergoing the first reaction when you're talking about the citric acid in the presence of an enzyme aconitase. It changes into, so this is an isomer form here. This is actually, it involves two-step process. One is your dehydration. Citric acid forming cis-aconic acid, okay, with the formation of your H2O, means it is a dehydration reaction. So the first step of, when you're talking about isomerization, it is dehydration, which is forming cis-aconic acid. Now this the product which is being formed, it is again undergoing hydration. Okay, cis-aconic acid plus H2O again in the presence of the same enzyme, that is aconitase, they are forming your isocitric acid. So isocitric acid is an isomer of citric acid. Okay, isocitric acid is an isomer of, that is your citric acid. So it is undergoing isomerization. So isocitric acid is also a 6-carbon compound. Okay. So it is a 6-carbon compound. Okay. So this is your isomerization here. Now you see, especially the isocitric which is being formed. Okay. The isocitric which is being formed. Okay. So there are two steps here now, which is actually demonstrating oxidative decarboxylation. Okay. It is demonstrating oxidative decarboxylation. So we can see that, okay, the first step, that is isocitric or isocitrate, okay, they are undergoing, again, there is a formation of NADH. So NAD, okay, so in the presence of dehydrogenation, so this process is actually dehydrogenation, okay. But in the last step, when you see, it is oxidative, isocitric is changing into, when you're talking about the intermediate form is, that is oxalosuccinic acid. Isocitric first changes into oxalosuccinic acid, that is actually oxidation, and oxalosuccinic acid finally changes into your alpha ketoglucanic acid. Okay, so oxalosuccinic acid, there is formation of NADH. Okay, NAD. Okay, they will be changed into NADH. Okay, and then oxalosuccinic acid, they are forming alpha ketoglucanic acid. It is undergoing decarboxylation. That's why the whole step here is being shown as oxidative decarboxylation. So isocitrate, if we, I'm writing here, isocitric acid. Okay. Isocitric acid, which was being formed. Okay. There is a formation of NADH from NAD. Thus, it is changing into oxalosuccinic acid. This oxalosuccinic acid, this was the intermediate step that I was talking about. It is finally changing into alpha ketoglucanic acid, and this alpha keto, this changing, it gives out CO2, and this is actually the oxidation process. Okay, so that's why we are calling it to be oxidative decarboxylation here. So at this site, there is a formation of NADH. So we should say we actually, during the process of Kreb cycle, there is a formation of, during the process of Kreb cycle, formation of 3-NADH. One step is here. Okay, one step is here, that is formation of NADH. Then again, when you talk about that is alpha ketoglutric acid. Okay, again it is changing into succinate CoA. Okay, again there is a formation of NADH. So this is also, so ultimately the first step of formation of is isocitric is changing into oxalosuccinic acid. Okay, then they are again changing into alpha ketoglutric acid. So that step is actually linked with the formation of NADH. And second step of formation of NADH is alpha ketoglutric acid, okay, changing into succinate CoA. Again, then there is the formation of NADH with the liberation of CO2. So it is also decarboxylation. Okay, with oxidation, decarboxylation is taking place because during the process of oxidation only, when the hydrogen atoms are being given off, then only this NADH is being changed into its reduced form. Okay, that's why we are saying oxidation. So oxidation of alpha ketoglutric acid, okay, changing into succinate CoA. So there is formation of NADH plus H plus as well as giving of CO2 here. Finally, there is also a combination of with the succinate. Okay, the coenzyme is being added. So then ultimately the product is succinate CoA. Okay, and the enzyme which is being taking place facilitating the reaction, it is your alpha ketoglutric dehydrogenase. So that is actually the enzyme which is related to the formation of succinate CoA. Especially when we talk about the enzyme, how to remember the enzyme, it is actually related to the product only. So suppose we are taking the example like this step only, like alpha ketoglutric acid changing into succinate CoA. So on the basis of the product, we have to name the enzyme. So the name of the enzyme is what? Alpha ketoglutric dehydrogenase, because when you're talking about it is process of oxidation, this removal of hydrogen is there. That's why it is dehydrogenation, removal of hydrogen, or rather we can say oxidation process. Okay, so it is your oxidation process. Okay, there is a formation of NADH. So that's why we are saying alpha ketoglutric dehydrogenase. In the previous process also, if we see isocitrate is changing into oxalo then to alpha ketoglutric acid, the enzyme is see the product on the basis of the reactant, especially isocitrate dehydrogenase, the second alpha keto dehydrogenase, because both the processes are related to removal of hydrogen here, okay, and addition. So there is the formation of NADH with the removal of CO2. That's why it is also oxidative decarboxylation here. Okay. Then the fourth step when you are moving, especially that is the succinate CoA, that is a very, very important step. We call it to be the substrate level phosphorylation. That is a very, very important step. The substrate level phosphorylation means it is the step where there is direct synthesis of ATP. There is formation of your GDP, which ultimately forms your ATP. Okay. That is monosine triphosphate, which changes into GDP, and GDP again ADP is changing into your ATP. They are actually the coupled reaction. Through the process of coupling of reaction, this GDP is again being converted into the ATP form. So this is the reaction, that is succinate changing into succinic acid, succinate CoA changing into succinic acid in the presence of an enzyme succinate thiokinase. It is actually the substrate level phosphorylation. Means there is direct synthesis of ATP. Okay, so if it is the only step in Krebs cycle where substrate level phosphorylation is taking place. So this is very, very important step. It is the only step where substrate level phosphorylation is taking place. At the rest of the cases we have seen, there are two steps where NADH has been formed. One, that is isocitrate changing into the alpha ketoglutric acid, and alpha ketoglutric acid changing into succinate CoA. These are the two steps where NADH are being formed. The two NADH have been formed. As told you in the beginning, in the Krebs cycle there are three NADH are being formed. Now we are going to see where they are being formed. One substrate level phosphorylation will be there, and again one molecule of FADH2 are being formed. So ultimately we have to see at which step these are being formed. These steps where they are being formed, they are very, very important. and the name of the reactions are very, very important. Why they are substrate level phosphorylation, which step is substrate level phosphorylation, which is your oxidative decarboxylation or rather dehydrogenation, So that is very, very important here. So this step is your substrate level phosphorylation. Now we are saying after the substrate level phosphorylation, okay, again there is oxidation with the help of, so if you see, okay, that is your succinic acid, not succinic, okay, it is your succinic acid. So succinic acid changing into fumaric acid. That step is again formation of a reduced coenzyme that is in the form of FADH2. Okay, that is your reduced coenzyme. So that step is formation of FADH2. So if you see the naming of the enzyme also, succinic, okay, so it is succinic dehydrogenase because this process is oxidation of succinic to fumaric acid. So at this step, we are having the formation of one molecule of that is FADH2, that is the reduced coenzyme. Okay. So this is another very, very important step where FADH2 are being formed. So this is the only step in the Krebs cycle where FADH2 is being formed. Only step. Okay, that is changing of succinic to fumaric. That is the step where FADH2 is being formed. Okay. So if we are moving from the cycle, if you see from citric acid, okay, we are moving to isocitric, then alpha ketoglupric acid. So alpha ketoglupric acid, if you see, okay, it is a 5-carbon compound. Why? Because in the first step only there was what? There was a decarboxylation reaction. Again, alpha ketoglupric acid is undergoing decarboxylation to form succinate COA. Okay, again it is your decarboxylation. Okay, so ultimately there is a formation of, if you see succinic acid and all, they are what? They are 4-carbon compounds. Okay, they are 4-carbon compounds. Succinate COA, that is also a 4-carbon compound. Succinic acid, which is being formed, it is also a 4-carbon. Because earlier decarboxylation, 2-step decarboxylation has taken place. Okay, so 2-step in the beginning also I told you when there was a link reaction, okay, the pyruvic acid changing into, that is your acetyl group, okay, acetyl COA, there was decarboxylation. So one CO2 was being formed there and 2 CO2 is formed here. So 2 decarboxylation has taken place. Okay, so 2 here, and when you're talking about 1 there, CO2 has been formed. Okay, so that has to be told to the students where CO, because in the beginning only I have mentioned 6 CO2 is being formed. And the cycle, especially one very interesting thing is that, especially in case of your glucose breakdown, that is glycolysis is taking once for complete oxidation of glucose, okay, to your CO2 and H2O. Glycolysis is a single step. Okay, why? The Krebs cycle is taking place twice. Because there is a formation of 2 pyruvate molecules. By the process of glycolysis, there is a formation of 2 pyruvate molecules. So when we are oxidizing glucose to form your ultimate oxidation is taking place where? It is taking place especially when you're talking about in the Krebs cycles only. Complete oxidation, suppose we talk about. Definitely the NADH, they are undergoing your oxidation, means in the ETC they are moving and they are going to be harnessed, the energy is going to be harnessed there. But ultimately the oxidation complete is taking place in the Krebs cycle itself only. So there is a formation of CO2 there. 2 in the Krebs cycle and 1 in the link reaction. So glycolysis is making 2 component, 2 pyruvate was there. So 1 glycolysis is taking place and 2 Krebs cycle is taking place. So altogether, these 2 and 1 pathway is completely oxidizing the glucose to your CO2. Okay, complete oxidation is taking place. So again we are coming back, that is your oxidation, that is a formation of your FADH2. Okay, formation of fumaric acid is there from succinic acid. So the enzyme again is your succinic dehydrogenase. Again this is being followed by the fumaric acid, which is a 4-carbon compound. Okay, it is undergoing hydration to form your malic acid. Okay, simply this reaction fumaric to malic, okay, it is hydration reactions. Okay, there will be addition of H2O and there is nothing formation at this step. turmeric to malic here. Again, there is a dehydrogenation reactions. Malic acid to oxaloacetic acid. That is again a 4-carbon compound. Malic acid is also 4-carbon. It is also undergoing, so there is formation of NADH, dehydrogenase. Malic D, in the presence of malic dehydrogenase, there is a formation of, that is oxaloacetic acid from malic acid here. So NAD plus is changing into NADH plus H plus ions. So there is formation of, that is NADH here. So ultimately, if you see, okay, there is the formation of yes. If we see this reaction, okay, so when we are beginning, that is your acetyl CoA, okay. If you see, it is a 2-carbon, okay. Formation of citric acid or citrate, it is a 6-carbon, because oxaloacetic or oxaloacetic acid, it is a 4-carbon compound which is combining with acetyl, so it is your 6-carbon. Again, isocitric or isocitric, it is again 6-carbon compound, because it is simply undergoing, when you're talking about through isomerizations and all, okay, it is forming isocitric, okay. Isocitric, again, it is going to form your alpha ketoglupric acid. Alpha ketoglupric acid, if you note here, it is a 5-carbon compound. 5C are there. Why? Because when isocitric is changing into alpha ketoglupric acid, there is a decarboxylation process. Single decarboxylation is taking place. So from 6C isocitric is changing into 5C alpha ketoglupric acid. Along with that, there is a formation of NADH at this step. Isocitric changing into alpha ketoglupric acid. Again, when we are saying this 5-carbon compound, when there is a formation of a 4-carbon compound, okay, that is succinyl CoA. So succinyl CoA, which is being formed, it is a 4-carbon compound, because again the alpha ketoglupric acid, it is undergoing, that is your oxidations, okay, along with when you're talking about decarboxylation. So when it is undergoing decarboxylation, if you see here, carbon dioxide is being released. Again, there is a formation of NADH, and there is addition of CoA. So that's why the product is succinyl CoA. Again, this succinyl CoA is changing into succinic acid. So succinyl CoA changing into succinic. Now succinyl CoA is how much carbon compound? It is a 4-carbon compound. Now from here, till when you're talking about the last, that is oxaloacetic acid, there is no decarboxylation. There is no decarboxylation. So there will be no reduction in the carbon number here. Okay? So there will be no reduction of carbon. So oxalosuccinic acid, till oxalosuccinic acid. So if you see, there is no decarboxylation. Okay, so from succinyl CoA, okay, which is undergoing. So succinyl CoA changing into succinic acid. It is as we have seen in the previous mechanism here, we have just discussed, there was a substrate level phosphorylation at this step. So there is formation of your ATP through coupling of reaction, that is your GTP. Okay. So there is formation, so one substrate level phosphorylation, formation of your NADH, 1, 2 we have discussed, one from isocitrate to your alpha keto to your succinyl CoA. So there is formation of 2 NADH here, and final formation of NADH is when the malic acid is being converted into, that is your oxaloacetic acid or oxaloacetate. Okay, so malic dehydrogenase. So again, on the basis of the substrate that we are using, we'll be naming the enzyme. So malic is there. Malic is to oxaloacetic acid. So these are the 3 steps where 3 NADH is being produced by 1 Krebs cycle, one cycle of Krebs cycle. Means one acetyl is being used, they are being oxidized, and finally forming your oxaloacetic acid, which is a 4-carbon compound. So what is the product? If you see, now we are seeing, if we are talking about the energetics of PCA cycle. So what we are saying, when this one acetyl COA, which is entering into the Krebs cycle, okay, ultimately one of these acetyl COA, they are forming one cycle. 3 NADH is being formed. 3 NADH is being formed. And I have mentioned the steps where they are taking place. Similarly, there is formation of 1 NADH2. So formation of 3, formation of 1, and the ultimately 1 SLP, substrate level phosphorylation. So through the substrate level phosphorylation, we are getting the ATP. That these are these two. So one acetyl, when they are undergoing oxidation, there is a formation of 3 NADH and 1 FADH. So they will be not providing direct energy. They will be moving to ETC. So if one acetyl COA is forming 3, so we have seen in the beginning, during the link reaction, there was formation of 2 acetyl COA. So if it is 2 here, okay, so ultimately we can say, if we multiply with 2 here, ultimately we are getting 6 NADH.
रात शाम में हमको फोन किया
Ultimately we are getting 6 NADH.
और फोन भी करने का समय बता दीजिए
Because the cycle will be moving twice, as I mentioned earlier. The cycle will be moving twice here. Okay. Similarly, there will be 2 FADH2,
गार्जियन लोगों से ज्यादा
FADH2, and we will be talking about
कैसे उनको पता चला कि आज
there will be formation. So one substrate level phosphorylation, so again there will be formation of 2 ATP there.
एक साइन अपना भी करते हैं सर.
हाँ
Because the cycle will be moving twice here. Because there is 2 cycles. One acetyl is forming 3, 1, 1. So 2 cycle will be forming 2, 6, 2, 2 here. And we know that, okay, 1 NADH. Okay, this NADH here, 1 NADH, if we talk about 1 NADH, they are forming 3 ATP.
कितना था? 0005.
They are forming 3 ATP. And 1 FADH, 2, they are forming 2 ATP. When they will be used, they are forming 3 ATP where? Using the ETC. In the ETC, when they are moved to the electron pump system, when they will be oxidized, okay, through the electron carriers, they will be releasing 3 ATP and FADH2 will be releasing 2 ATP. We'll be seeing with the ETC, okay. So 3 ATP and 2 ATP. So ultimately, if you see, so 6 NADH was being formed. So if we 6, so we are getting 18 ATP. And here it is 2. So it will be 4 ATP. And 2 direct ATP is there, so 2 ATP. So ultimately, if we talk about, there will be 24 ATP
आज सुबह मेरे क्लास का था
which is being formed. 2 ATP through your substrate level phosphorylation. So altogether, if we see the potential of Krebs cycle, it comes with your energetics aspect here. How much energy potential? It is 24 ATP. Okay, so 24 ATP by means of your Krebs cycle. Definitely, they are not the direct form of ATP from the Krebs cycle. When they will be moving to the ETC, then only will be getting 3 ATP and 2 ATP respectively from 1 NADH and 1 FADH. Okay, so that has to be mentioned here. So they are not. So they are also the energy sources. Okay, they have stored energy. But when these reduced form of coenzymes, when they are being moving along the electron carriers, they are giving out the electrons along with the protons they are giving off, and that is being used for harnessing the energy which is being locked in these reduced form of coenzyme. So they also have the energy source, but they are not the direct source. When they are moving along the electron carriers which are present in the respective complexes of the electron transport system, then they will be giving out this 3 ATP and 2 ATP respectively. So the Kreb cycle, ultimately, if we talk about, we are having 24 ATP. If we link with the link reaction, okay, so we saw one pyruvate, they are giving one NADH. So ultimately, there will be formation of 2 NADH there. Okay, in the link reaction, I am talking about in the link reaction, that is conversion of, if we see, yes, if you see here, this is the link reaction. The pyruvic acid changing into acetyl, so they are also forming NADH, one from one pyruvic acid. So there are 2 pyruvic acid which is being formed by the means of glycolysis. So ultimately there will be formation of NADH here. Okay, so 2 NADH is also being formed through your link reaction. Through the link reaction. So 2 NADH they will be forming, they will be giving rise to how many ATP? That is your 6 ATP. 6 ATP. So 24 plus 6, if you see, it is coming. How many? That is 30. So where is the 8 here? 8 is from the first step that we are talking about, that is your glucose which is changing into pyruvate. Then also when we are talking about 2 ATP is being formed in case of glycolysis. If you talk about glucose changing into, that is your pyruvic acid or pyruvate. There is a formation of 2 ATP by substrate level phosphorylation, and there is also formation of 2 NADH there also. So 2 here and 6 again from here, because I have mentioned here 1 NADH is giving 3, so 2 again here will be 6. So 6 from here and 2 ATP through your SLP, that is substrate level phosphorylation. So ultimately we are getting, that is your 38 ATP. But in some cases we see, especially it is your 36 ATP. Like in case of your muscle cells and all, we ultimately the total energy if you see, it is 36 ATP, because 2 ATP are being used. Ultimate production is definitely 38. 24 from here, 6 from your link reaction, so 30. Then we are getting 8 from your glycolysis. This process glycolysis. 8 from we are getting from here. So ultimately, if you talk about the energetics at the total level, complete oxidation of your glucose into your CO2, H2O, so then if you're talking about the energy, it is 38. But there are 2 ATP which are being used for the transport of what? This pyruvic acid. This process, glycolysis, is taking place where? It is taking place in, when you're talking about cytoplasm. While the Kreb cycle, it is taking place where? In the mitochondria. The link reaction is also taking place where? It is taking place in mitochondria itself. So for this process, okay, to take place, the pyruvic acid which was formed in the cytoplasm, it needs to move inside the mitochondrial matrix. So for the movement, for the transport of this pyruvate or the pyruvic acid into the mitochondrial matrix, there is expenditure of 2 ATP. So then we come to 36 ATP here. So that is very, very important that we tell the students here. Ultimate it is 38, but it is 36, especially like in case of your muscle cells and all, it is 36 because 2 ATP are being used for the transport of pyruvic acid to the mitochondrial matrix. Okay, so that's why there is 2 ATP is lesser, because that has been used for the transport of pyruvic to where they will be transported from cytoplasm to mitochondria, and mitochondria, especially the matrix, because the Krebs cycle is taking place in the mitochondrial matrix. The link reaction is also taking place in the mitochondrial matrix. Okay, so ultimately, what we are seeing, in terms with the energy, the whole oxidation process, when you're talking about of your glucose, we have started with what? We have started glucose here, that is C6H12O6. So this complete oxidation of 6 carbon, we are requiring 6 oxygen, and ultimately there is formation of 6 CO2 plus 6 H2O plus your energy, and that is the energy that we are talking about. So 6 CO2. Again, I am talking about, so this CO2, it is not being formed in pyruvic acid. It is only in the link reaction, as we have discussed earlier. Decarboxylation reaction was taking place. If you see, there is a decarboxylation of pyruvic acid changing into acetyl. So it is decarboxylation 1, okay, is formed there. 1 as when pyruvic acid is being changed, 1. So 2 will be formed here. And 2 are formed. If we are talking about in the Krebs cycle, we are forming all together because it's a 2-step process. So all together there will be formation of 4 CO2 there. So all together we are having 6 CO2 which is being formed here. And complete oxidation is taking place in Krebs cycle here. So this is all about when you're talking about the Krebs cycle process. Okay? This is all about. So what is the significance of the Krebs cycle if we talk about? The significance of the Krebs cycle, because when you're talking about glycolysis, it is a partial oxidation. Partial oxidation of glucose. Okay. But if you talk about the Krebs cycle, there is complete oxidation of glucose. Means we are able to harness the whole of the potential of the glucose in terms with the energy aspects. Okay. In terms with the energy aspects, we are able to harness the whole potential of the glucose by means of your Krebs cycle. Okay, and that's why we are getting, if you talk about the Krebs cycle, okay, the products which are being formed, they are ultimately definitely giving rise to 24 ATP. Okay, in the Krebs cycle, if we take into account link reaction, which is also taking place in the mitochondria, it will be 30 ATP. Definitely, it is only, if we talk about direct ATP, so it is very, very less. It is very, very less. Suppose we talk, now how we relate Krebs cycle with ETC. How we are relating Krebs cycle with ATC. Talk about this aspect. Suppose so if we are talking about direct ATP through your substrate level phosphorylation. So ultimately what we are seeing from glycolysis, if you see, okay, it is your 2 ATP through substrate level phosphorylation. And through the Krebs cycle, I am talking about direct ATP through substrate level phosphorylation. Through your Krebs cycle, it will be 2 cycles. So 2, when you're talking about 1 cycle substrate level phosphorylation, only 1 ATP is being formed. So 2 cycle, again 2 ATP. So ultimately, we are getting how many ATP? It is your 4 ATP only. Means the glucose, okay, if you're talking about direct ATP synthesis through your substrate level phosphorylation, it is only 4. When the hidden potential of the glucose in terms with the energy, we are not able to harness if it is not through your electron transport system. If ETC was not there, why we are moving to ETC? What is the significance of the ETC here? What is the significance of what is the purpose of ETC? If you talk about the purpose of ETC, what is the purpose? What is the main purpose? Why we need to go to ETC? That can be also the question. Why do we need to? Because the purpose of ETC, first, when you're talking about, we are saying there is a formation of NADH plus H plus. These are the reduced form which are being formed in your link reaction, in glycolysis, in your Krebs cycle also. So if the cycle, we are saying that the cycle need to be continued twice, especially when you're talking about the Krebs cycle or the link reaction. They are twice process. So if needs to be continued, we need to have what? We need to have the presence of NAD plus. If this is not moving to ETC, NADH is not moving, or the reduced form is not moving to ETC, we cannot reload NAD plus, okay, in the matrix. We cannot reload this NAD plus in the matrix of the mitochondria. And there the significance of ETC. What will happen here? So this NADH, okay, this NADH plus H plus, they are undergoing oxidation in your ETC. So there will be formation of what? NAD plus, plus 2H plus, plus 2 electron minus. So this is what is happening in case of your, when you're talking about ETC. So there will be, this is what? This is again being reload where? In the matrix. And if it is being reload, again the Krebs cycle will be taking place. If it is not being reload, suppose the NADH, they are not moving to the ETC, we cannot have NAD plus reloading of NAD plus. So the cycle, that is the Krebs cycle, will be stopping. Okay, that is very, very important. This is the significance of your ETC, the first significance here. Reloading of NAD plus through the oxidation of NADH. The second is very, very important purpose is oxidative phosphorylation. Oxidative phosphorylation. Oxidative phosphorylation, which is actually the synthesis of ATP. The synthesis of ATP. So ETC, when they are being oxidized, okay, there is your proton, there is an electron. So these electrons, they move along the electron carriers. The electron transport system, they have different complexes. Okay, so complex 1, 2, 3. Okay, there are 4 main complexes along with the 5th complex, which we call them as ATP synthetase. Okay, but there are more 4 complexes here. Okay, but 3 are very, very actively involved. Okay. So there are 4 complexes. So these 4 complexes, they have the presence of, if you see, these are the complexes here. Complex 1, complex 2, complex 3, and complex 4. These are the 4 complexes. Electron transport system. So the purpose is oxidative phosphorylation and reloading of NAD plus, so that the cycle can continue. Especially it comes with the reloading. So when you talk about ETC, ETC is taking place where Krebs cycle is taking place in the mitochondrial membrane. ETC is taking place where in the inner membrane. The inner membrane. Okay, they are taking place in the inner membrane of mitochondria. So inner membrane of mitochondria. And this inner membrane of mitochondria, they are having these complexes. These complexes, they are present along the inner membrane of mitochondria. And these complexes, they have the presence of specific electron carriers. They have the presence of specific electron carriers. So this, as we have discussed here in the previous class, that is NADH plus H plus, when they are being oxidized, there will be formation of NAD plus 2 H plus 2 electrons. So this electron will be passing through these electron carriers, and especially these complexes. Very, very important here. These complexes are being arranged along the inner membrane of mitochondria in terms with their redox potential. They are being arranged in terms with their redox potential. Means from higher to lower. Means the complex one will be having higher redox potential. Means they will be having a negative value. Means they are in the form of reduced. So they are higher redox potential. They have the presence of higher redox potential. Very, very important. And as we are moving from complex 1 to the complex 4, they are having lower redox potential. So that's why the electrons which are being moving along these complexes which are having these electron carriers like FLN, FES, which is present in complex 1. Complex 1, they have the presence of these electron carriers. Okay, so it is also known as NADH dehydrogenase complex. Okay, this is your electron carriers. Again, we are having another electron carriers as ubiquinol, UQ. Again, it will be, so this is when talking about your mobile carriers, which is again related to the other complexes, that is your complex 3. Okay, so this, first I'm telling you what is the significance, how it is moving. So the inner mitochondrial membrane, they have the complexes, and these complexes have the presence of electron carriers, and these electron carriers facilitate the movement of electrons from one complexes to the other. So as the electrons are moving from higher redox potential to the lower redox potential, they are giving out their energy. They are giving out their energy, and ultimately, as they are moving, the electrons are moving from the electron carriers. As they are moving, ultimately they are coming to whom? Ultimately they are coming to where? To the complex 4. Okay, to the complex 4 especially, if you see, so electrons are coming. Okay, there will be some formation, there will be also the protons which are coming, plus your half O2 will be there, and ultimately will be having what? Formation of your H2O. And this formation of, this is actually, we call them as terminal reaction. This is the end of the terminal, means your electron transport chain reaction here. So this is your terminal reaction which is being. So the electron which is being passing from higher to lower, ultimately they are being picked up by whom? They are picked up by the oxygen. So ultimate acceptor of electrons as well as the ultimate acceptors of the proton, it is your oxygen. Means if oxygen is not being available, then they will be not accepted and the ETC will be stopping it. Okay, so they are the ultimate acceptor, and that's why there is a formation of your H2O. So they are accepting, so there is a formation of this water. This is also known as, when we are talking about metabolic waters here. Okay, so this is terminal reaction. Ultimately, when there will be, the electrons will flow along these electron carriers here. Okay, and should I continue? Yes, sir. Hello.
Yes, what do you want to ask? You finished? Raju sir, I cannot understand what you are asking.
That is your NADH plus H2O. And here, this NADH, we are dividing into your complexes. This is your complex 1. Okay. This is your complex 1 of inner mitochondrial, where we are talking about the ETC here. Okay. So this is your FMN along with your FES. Then again we are having this ubiquinol. Again we are having another complex, that is complex 3, that is your cytochrome PC1 complex, which is also having the electron carriers as FES, and leukoquinone. Again, we are having the electron carrier, that is cytochrome. Okay, cytochrome C. Actually, when we're talking about the electron carriers are in the form of FMN, FES, and cytochrome. And if you talk about the cytochrome, which is actually your iron proteins. Iron protein complexes are there. Okay, and there are actually 5 types. That is your B, okay, C1, then we are having C, A, and A3. So these are the 5 cytochromes. Okay, and this cytochrome A3, this is a very, very important. Okay, this is cytochrome oxidase. And this cytochrome is a very, very important one. Okay, because ultimately, okay, they are picking up when you're talking about with the oxygen. So A3 is also related to carbon monoxide poisoning or cyanide, especially cyanide poisoning. It is related to cytochrome A3. Okay, so there are 5 complexes here. That is B, C1, C, A3. So they have been arranged in this form along with the complexes. So this is again your mobile carriers. Again we are having, this is complex 4. So the complex 4 is cytochrome A along with your A3. This is your complex 4. Here when we are talking about, there is an association of complex 2 here. Complex 2 is associated and it is your FAD along with your FES. Okay, and here we are having complex 5. Complex 5 as your ATP, it is like this. Okay, this is F0, F1, and there is actually formation of ATP is here. This is your ATP synthase, and this is your F0, F1 particles, which we tell the students structurally, especially in mitochondria, we have the presence of elementary particles, that is oxysomes. So F0, F1. So what is the role of F0? It is acting as proton pump. इसी through this only, the protons are being pumping. Then suppose this NADH, this is complex 1, so they are taking it. So NADH, they are again being reloaded. NADH, the equation we have done, it is your NAD plus 2H plus 2 electron minus. So this H is being taken by FMN along with the electrons, which is taken by the electron carriers. So this FMN, if they are taking FMN, if they are taking this 2H plus, it will be again reduced to FMNH2, and again they will be converted, oxidized here. So this is being taken here. If you see, if I remove the complexes, now you see here, they are being converting into what? FNH2. Okay, if we talk about, so they are being converted in the form of FMNH2 here. Okay, and this proton, again they are being changed into what? FMN. Again they are being changed into FMN. So this proton is again being released. So they are being pumped where? They are being pumped where? This is what? Perimitochondrial space. So from the matrix, they are being pumping. The proton is being pumped where? Perimitochondrial space. Perimitochondrial space. This is the inner membrane, and this is your perimitochondrial space, and this is your outer membrane, and this is your cytoplasm. This is your cytoplasm. This is your outer membrane. This part is your outer membrane of mitochondria. This is your perimitochondrial space between outer and inner membrane. So here the protons are being pumped. So as the protons are being pumped from the matrix, we have seen there is a formation here. As they are being pumped, there is a difference in the proton gradient. Okay, so now again they will be pumped. This proton, again they will be pumped back to where? Back to the matrix. So if they have been pumping, suppose this proton is being pumped through these, that is your proton pump, that is your oxygen particle. Again, they are being pumped. Suppose the protons are being pumped here. So as the protons are being pumped, they are giving out their energy. So that energy is being used for synthesis of your ATP using your ADP and inorganic phosphate, utilizing the complex fifth, and that is your ATP synthase. This is the complex, the last one. Okay, so this is your ATP synthetase complex, which will be utilizing this proton, which is being pumped in the mitochondrial space again into the matrix here. Because, and if you see, the electrons will be picked up by here. So this Fe, जो 3 है, ये क्या में convert हो जाएगा? Fe2 plus में change हो जाएगा. Again, they will be converted back to what? Fe3. So again, the electrons will be given to whom? To here. अब जो proton pump इधर में हुए हैं, and there is again they will be taken by the ubiquinone and ubiquinH2 will be there. Again, it will be pumped here. Again, it will be pumped back to the mitocat here, and again they will be taken by whom? The complex, that is your 3. Okay, again they will be reduced, taking the H2 here. So at 3 steps we are taking. अगर simply अगर हम लोग बनाएं इस तरह से, suppose we are talking FML, SES, ubiquinone. It is related to the site, means complex 3 अगर हम लोग देखें, suppose the complex 3 is there. Okay, this is your cytochrome BC1 complex. This is, so protons are pumped from here. Okay, protons are pumped from here. Protons are pumped from here. यहाँ जितने भी pumped हो गए protons, they are being pumped here. So how many are pumped here? 3. 3 are being pumped. So 3 are generating ultimately ये क्या होंगे? Using this, that is your ATP synthetase, that is the proton pump, oxysomes. ये 3 बार हो रहे हैं. Means what? There will be synthesis of 3 ATP. And who is being used? NADH. So when we are saying in the Kreb cycle, 1 ADH is giving how many ATP? 3 ATP. So these are the 3 ATP which is being synthesized. And FADH कहाँ पे enter करेगा? इस side पे, ubiquinone वाले जो side, this side FADH2. FADH2 will be not entering into the FES complex, that is complex 1. It will be entering at this side. Why? Why the FADH2 is entering at this side? Because the FADH2, I have told you, these electron carriers are being arranged according to the redox potential. Means the complex 1 is higher redox potential, complex 4 is lower redox potential. So FMN, okay, FES, they are higher redox potential, the complex. FED and NADH plus H plus, they are present at this complex 1 site, higher redox potential. That's why they are moving from first using ubiquinone to third. Okay, FADH2 will be moving along only these 2 sites. That's why 1 FADH, because FADH2 का redox potential is accordance with the ubiquinone and इस site के accordance उनका redox potential. That's why FADH2 is entering at the second site, not at the first site. Okay, that's why they are only giving 2 ATP using ubiquinone and cytochrome when you're talking means rather the complex 3 here. So ultimately 2 ATP and 3 ATP is being formed here. Okay, thank you.
Thank you. Thank you, Raju sir. What we felt right now, he tried his best, he explained very well. Had it been an offline session, it would have been enjoyed more by all of us. Isn't it? His blackboard board, the way he explains is excellent. We would have enjoyed it more had it been an offline session. Sometimes what we were writing on the blackboard was not visible, otherwise it was very, very well conducted. He explained everything. We're sticking to Kreb cycle and electron transfer chain. But sometimes you include where it happens in between so that it's not practical. Just please discuss.
Am I audible? Alok sir, am I audible?
Absolutely, sir. Clear.
Raju sir explained this topic brilliantly, no doubt.
Not able to hear. Yes, sir.
Now I think it becomes. Is it? Am I audible now?
Yes, you are audible.
Raju sir, you explained your topic brilliantly. I personally enjoyed a lot.
Yes, sir.
I would like to highlight.
Hello.
I would like to highlight on 2 to 3 main points. When you are explaining link reaction, that was oxidative decarboxylation. We know that 3 carbon compound, that is pyruvate, cannot penetrate the inner mitochondrial membrane. So pyruvate has to be transferred into acetyl group. So as we know, during link reaction, pyruvate is converted into acetyl coenzyme. Before acetyl, actually acetate molecule is formed. So 3 carbon compound is converted into 2 carbon compound. Then it gets entered the inner mitochondrial membrane. That was brilliant. Second, whenever there is any kind of dehydrogenation state in cellular respiration, whether it is glycolysis or Kreb cycle or citric acid cycle, what happens normally? A pair of hydrogen ions are released. That hydrogen ions are accepted by coenzyme. It is either accepted by NAD or by FAD. NAD accepts hydrogen, gets reduced to form NADH plus H plus, which are oxidized, which are again oxidized. Actually, the redox reaction is going on. So it passes down the electron transport system, and as a result of oxidative phosphorylation, whenever there is oxidation of reduced coenzyme, energy is formed. And you are explaining synthesis of ATP. Whenever there is complete oxidation of glucose molecule, in NCERT book it is written that when one molecule of glucose is completely oxidized, 38 molecules of ATP are formed. We know that there may be formation of 36 molecules. Actually, there are 2 types of shuttle systems. One is malate shuttle and another one is glycerophosphate shuttle. Actually, malate shuttle is considered in NCERT book. That is why 38 molecules of ATP are written over there. But you have already explained in your topic 36 in case of muscle cells. That was I liked much more. So thank you very much from my side. And in cytochrome, that is a special conjugated protein complex, involvement of iron sulfur complex. You also explained that point. I liked very much. Thank you very much, sir.
Thank you, sir. Thank you. Thank you, sir.
Yes, any question or any query or any suggestions? Any participants can come forward.
Yes, Raju sir.
Yes, sir.
Krebs cycle is the one of the mechanism of the cellular respiration, and you have built their mechanism as well as different steps of the mechanism with the help of chemical reaction as well as in the form of cyclic form. That is amazing.
Thank you, sir.
Always a scope of discussion among the student in a well way, just like net reactions we have to discuss. What is the total number of net gain of total number of NADP, FAD, in which circumstance malate, Dubey sir has already told, malate already plays significant role. When NADH2, FSH, FADH2, these all things very important. And whenever we are discussing cellular respiration, glycolysis, that is EMP pathway, Kreb cycle, electron transport system, we have to conclude together, because one mechanism is linked with another one.
Yes,
Suppose
sir, yes.
36 ATP, what is total net gain in case of EMP pathway, that is glycolysis? In one case 8 ATP, in another case only 2 ATP. What is the total number of substrate level oxidative phosphorylation? What are the sites? These all things are very important, because during entrance examination such type of questions are always asking.
Yes.
That's why we have to make the keynotes regarding such type of the things. So whenever we are delivering the lecture in the classroom, then in the form of key points, these all things when we will discuss on the board, then it will be very useful for the students. Right, sir?
Thank you, sir. Thank you, sir.
Raju sir, thank you.
Thank you, ma'am. Thank you.
It was really again revisiting all the topics, and sir has nicely pointed out these topics are a bit difficult for the students to understand in one way or one go. So mind map sort of, or short definitions, or one topic at a time, that really helps. So thank you, sir. It was well defined and well explained. Thank you
Thank
once.
you. Thank you, ma'am. Thank you.
Teachers, I have shared a few pages to make it concise. What you were speaking, it will remain with you. You can always go through that. On the group, I have shared a few tables for the ATP generated and a few pages. On the WhatsApp group, I have clicked a few pages and said, if you will read that, it becomes very easy to understand. Okay, I have also shared the attendance link. So there is no question. You may continue asking and then fill the link to the attendance and assessment as well. Yes. Thank you, everyone. Thank you, Raju sir.
Thank you, ma'am. Thank you.
Thank you, ma'am.
Thank you, Raju sir.
Thank you, sir. Thank you.
Very nicely explained, sir. Very difficult to deal each and every point in such a short time. Really,
Thank you.
hats off to you.
Yeah.
Despite
Thank
of
you.
all technical constraints, you have managed it so well. And thanks to
Thank you.
your helpers,
Thanks to
your
all
friends
the
also.
teachers also, they have cooperated. Thanks to all the teachers.
Yes, certainly, sir. Certainly.
It was just like classroom experience, Raju
Yes,
sir.
Yes, sir. Yes,
It
sir.
was
Yes,
the
Mr.
practical
Raju.
classroom.
Yes, sir. Yes, sir.
Thank you very much for your excellent presentation. Very nice.
Thank
To add that had it been offline, it would have been more wonderful, isn't it? It would have been 10 on 10. Right?
Yes,
now,
yes,
it's
sir.
8 on 10 because in this case they want to step forward. But then later you picked up very well, and you should say very well. Thank you, sir.
you, ma'am.
Mr. Alok, I think another break is there, na?
Yes, sir. Thank you, sir. So we'll sign off, ma'am, right now and join for the next session.
We'll be joining 11:10.
Yes, sir.